So they're not alive. Period. For my own purposes, I must say sentimental purposes, want to think about them as living organisms because they extremely sophisticated, but they do lack certain properties that define a living organism. However, I insist on calling them microorganisms because they hijack cells very successfully. How many species? We have no idea. The estimate of 1 million is as good as any other estimate. There's really no way to estimate. We do know that so far, less than 300 viruses were identified that cause human disease. Just that's it, out of all the species that can be found. They must have a cell to reproduce. Okay? And the reason why we don't consider them living, for the most part, they don't have cellular structure. They also do not have their own ability to produce ATP. So they use the cellular ATP. Viruses also don't have ribosomes, so they cannot carry out their own protein synthesis and depend on the cell for this. Structurally, viruses are divided, and there's a lot of classifications. The problem with classification of viruses is that what makes a lion and a tiger two different species? Genetics, yes, but okay, let me ask you this. Does anyone have a dog? Can somebody tell me the breed? Okay, we have Jack Russell. We have Jack Russell Terrier. We have Cocker Spaniel. Different breeds? Yeah? Look different? Can they have babies? Can those babies have their own babies? That's the answer. That's same species. Because they produce fertile offspring. A lion and a tiger will produce a liger, which is a hybrid, and hybrid is infertile. It's a beautiful animal, but infertile. But how do you define species in organisms that are not even alive? There's a bunch of classifications. Mostly the classification is based on genetics, but the functional classification is here. Naked versus envelope. So this is a naked virus, one of many. As you can see, it has capsid. Viral capsids are made of proteins, always. And the genetic material. We'll get to the genetic material. Okay, so basically, genome. Or viruses can be enveloped. So just like naked viruses, capsids are made of protein, but outside of the capsid is the envelope. Envelope is phospholipids, which viruses steal from a cell, specifically from membranes, and various viral proteins. Okay? So envelope, the phospholipid layer, is relatively labile. It's fragile. So enveloped viruses are fairly easy to inactivate. Don't remember if I brought this up, but if you have a kid and the kid has flu and sneezed and coughed and all the toys, and you're concerned about the secondary transmission, put the toys in like a cupboard for 5 days. Virus will degrade by itself because influenza is enveloped virus. On the other hand, if you will try to do it with the rhinovirus or norovirus, good luck with that. They are naked, they don't have that fragile envelope, so they don't care. The first discovery, Ivanovsky, discovered tobacco mosaic virus, plant virus, since the virus was coming through the filter that was supposed to contain infectious agents. He called it infectious agent, filterable agent first, and the term viruses came much later. Viruses can be very small, such as polio. Hepatitis D, I think, is the smallest virus, even smaller than that. Or they can be gigantic. Pandora virus can be seen in the light microscope. It's so big. And it was discovered only in early 2000s because nobody thought that this big-ass structure is a virus. Okay? People saw it before. They just could not think of it as a virus. Okay? Now, there are a lot of classifications. Some of that terminology remains. For instance, we use arboviruses, the term for the virus that are arthropod-borne, arbo. Okay? But the true classification is based on the replication strategies, namely in the genome. And I want you to be aware of what genomes viruses can have. They're not alive. They don't have to be stuck with a double-stranded DNA. So it can be DNA, it can be RNA. It can be double or single-stranded in both cases. Now, what's the difference between the positive and the negative single-stranded? Well, here's the thing. Listen, if you have RNA, can it undergo translation? It can. You can translate it into a protein. Okay? RNA that can be used as a template for protein synthesis is positive. That's a definition. Okay? Now, what is a negative? So imagine a virus has a negative stranded RNA. It has to copy it using the principle of complementarity, just like DNA replication happens. Does that make sense? I would not even call it a replication. I would call it some weird transcription. So, negative stranded RNA viruses, such as Ebola virus, have a special enzyme in them that will convert the negative stranded RNA, negative single-stranded RNA, into a positive one, and that positive one will be a template for protein synthesis. But the genome is negative. Okay? Positive, my personal thought, positive stranded RNA viruses are the best. They're most interesting, they're very dangerous, they're cool. Okay? And in terms of studying, you can, let's say yellow fever. You can isolate RNA from the yellow fever. You can stick it in the cell. There are ways to just force it, like push it inside the cell. And the cell will translate this RNA and will make a virus. Viruses self-assemble. Okay? With the negative stranded RNA, you cannot do that. Structurally, I probably gave you plenty of examples where scientists become lazy in terms of naming. So viral capsids can have different shapes. The most common shape is icosahedral shape. The easiest way to think about it, think about soccer ball. That's as close as it gets. Kind of, sort of spherical, sort of, you know. So these are most common. There's a fair amount of helical capsids. Okay? Do you see that? Like a helix. Pretty simple. When the capsid has a weird shape, we're going to call it complex. It can vary. Smallpox has a very complex capsid. Rabies has a capsid that has a shape of a bullet, literally. Filoviruses such as Ebola have filamentous capsid, so, you know. Now, the separate story is bacteriophages. They do have a capsid that sits on top. Now, not all bacteriophages are like this. This is they call T even, nobody cares, whatever. So there's a certain group of bacteriophages that look like small alien ships, you know. There's a capsid, there's a tube, there's a sheath that covers that tube, there are fibers on the tail that bind to the cell, and these are one of my favorite structures. These are tail pins. Bacteria, do they have cell walls? Many of them do. So for a phage, it's not easy to get in the cell. These phages use tail pins to drill a hole in the bacterial wall. Okay? Now, when phages infect, and I'm going to stick to this statement, bacteriophages infect bacteria and archaea. I prefer to use bacteriophages for all of them. People who study those will probably kill me for doing this because they want to distinguish between viruses that infect bacteria and viruses that infect archaea. But whatever. So bacteriophages come in two flavors. They can be lytic and they can be lysogenic. We already talked about it. So lytic phages, they attach to the bacterial surface, and then during penetration, the phage DNA, not the entire thing, but only DNA, enters the cell. During the process of biosynthesis, that DNA is being transcribed, RNA translated, new phage particles are assembled. During the maturation stage, DNA gets itself into the capsid, and during lysis, the phage leave the cell. And effectively, bacterial cell is dead at this point. Okay? Now, since phages can kill bacteria that efficiently, the idea is that we can use those phages as the antibacterial therapy. Okay? So, problems, right away problems. I'm going to tell you problems and then the success story. Problems. When you develop a phage that kills, I don't know, salmonella, how do you know that it doesn't kill anything else? If you develop a phage that kills salmonella, salmonella will be co-evolving with the phage. It's a living system. It's a dynamic system. So there's a fair amount of resistance. And last but not least, phages are made out of protein, by and large, which is not natural for your body, which can be considered foreign. There's a non-zero chance that your body will develop immune response to a phage. Not that you're going to get sick, it's going to be antibodies. And if you try to use it again, the immune system will shut it down. Those are very legitimate concerns. But here's the success story. Again, real story. If you want to listen in details, not my very short rendition, I can give you the link to the podcast. Okay? So, bless you. A husband and a wife, both professors from University of California, San Diego, travel in Southeast Asia, where the husband gets exposed to bacteria called Acinetobacter baumannii. This bacteria, if it gets in the circulatory system, is bad. So by the time they go back to the States, he's very sick. He ends up in a hospital, septic. So septic that he has like wounds with pus coming out of them. He's comatose and it's totally drug resistant. So he's given a few weeks to live. Okay? Pretty bad. So his wife, she's a professor, not a biology professor, but she's in higher education, so she knows how Google works. And she finds a team of scientists at University of Texas who do phage therapy. And basically tells them, do you want to try to save my husband? They cooperate with a lab that is under Navy. And these two labs, and the guy said in the interview, they were literally working 24/7. So the efforts of both labs completely were focused on developing and growing the phage that would kill that Acinetobacter. They delivered a huge can of the phage solution and started IV on that guy. And 2 weeks later, he walked out of the hospital. Yes, he was cured. And they had another story. They had a patient in Texas, it was Austin, Texas, in Texas, who had fistula. And do you know what fistula is? Yeah, like a hole in the body that doesn't heal. So the fistula, which formed after he got an aortic replacement. And it was infected and never healed, and like you walk around with a hole in your chest. Not great. So those folks thought, what are we going to do? How are we going to cure it? And finally, they decided to just put the phages in the syringe and just spray the fistula. It healed. So the problem is, again, you have to have a good library of phages. You have to screen. You have to be careful. Okay? But it's kind of getting momentum. Lysogenic phages, we talked about them when we discussed specialized transduction. So you can see that the phage gets incorporated into the bacterial chromosome, stays there, cell reproduces so it doesn't have to put any effort in. If the cell becomes stressed, the phage cuts itself out, starts producing new particles, and then kills the infected cell, lyses it. Okay, so it's a pretty selfish strategy, right? So you get in the cell, and while everything's great, you're quiet, but when the cell becomes stressed, the phage not just gets out, it kills the bacteria. Okay? Lysogenic, because they can potentially generate the lysis. So what we're going to do, I'm going to finish like the general considerations for the viral infection, and then we're going to move to the lab. So eukaryotic infections. I really want to ask people who wrote a really good book on microbiology, textbook, why they used influenza. They used an exception to illustrate a rule, which is not great. So I will try to amend this as much as I can. First of all, what do viruses need to get attached to the cell? That's not a rhetorical question. A receptor. Do you think that the receptor that they attach themselves to, do you think that this receptor is just a unique molecule that can only interact with viruses and nothing else? No. They use some functional protein that has some other role in the cell as a receptor. We talked about HIV. Its receptor is CD4 molecules on the surface of CD4 T cells. Okay? There's I believe it's a glycoprotein called DC sign that found on, it's an acronym, that's found on dendritic cells and many other cells. some viruses, especially flaviviruses, readily use DC side. Okay? What's important is that if cells have a receptor, they're called susceptible. If they don't, they aren't susceptible and cannot be infected. Does it make sense to you? That will become very important when we start talking about different viruses, and I will illustrate that receptors basically define where the infection is going to happen, which cells are going to get infected. Also, imagine you are running from a bunch of zombies, and you know that in a week, the apocalypse, well, 2 weeks, the apocalypse, zombie apocalypse is going to end. They're all going to be killed, but you need to survive for 2 days. And you run and you have a key in your pocket, and there's a door, and you open that door, and you run into the room, and you lock the door behind you. You're in the cell, but there's nothing to eat inside. So, for the virus to replicate, entering the cell isn't enough. The cell should support viral replication. Cells that support replication are called permissive. Can a cell be susceptible but not permissive? Yes. It can get in, but it may not replicate. For example, there was a publication in the beginning of COVID pandemic, when people already knew that SARS-CoV-2, the virus, infects lung epithelium, sure, okay, fine, infects and reproduces, but the virus was also found in the endothelial cells in the blood vessels. This publication the study was done by doctors. When virologists saw it, they critiqued it really hard, because they never showed that virus actually reproduces in those endothelial cells. It does enter, but that means nothing really. Make sense? So, for the full infection, cells must be susceptible and permissive. Okay? So, after the attachment, the entire virus, unlike in case of bacteriophage, the entire virus gets into the cell. How? Here, authors suggest endocytosis. It can also be so-called fusion, when the envelope of the virus, which remember is pretty much a membrane, fuses with the cell membrane and releases the capsid into the cytoplasm. Okay? Then there is a step that is not present in bacteriophage infection, specifically uncoating. The capsid has to be disassembled to release genetic material of the virus. Right? This is a mechanistic process, usually triggered by changes in pH. So now, if we're talking about RNA virus, and RNA is in the cytoplasm, does it necessarily alarm the cell? RNA in the cytoplasm. What do you think? No, not really. I mean, it will eventually, but let me rephrase it. Is RNA, garden variety RNA, in cytoplasm normal? Yes. Ribosomes are in cytoplasm. It's fine. Is DNA in the cytoplasm normal? That's not. There's a whole system. Remember, we're talking about pattern recognition receptors. There's a whole system called GAS-STING, which is a long acronym, GAS-STING. Its function is to sense DNA in the cytoplasm and send a cell into apoptosis. Okay? So, for the most part, there are exceptions. RNA viruses they reproduce in the cytoplasm. DNA viruses reproduce in the nucleus. So that allows them to kill two birds with one stone. They can hide DNA in the nucleus, right? And they can use some of the DNA replication enzymes that the cell uses, or even transcription enzymes, because all of that happens in the nucleus. Okay? Why I said that the use of influenza is kind of weird. Influenza is RNA virus, but it reproduces in the nucleus. It's an exception. There is a DNA virus that reproduces in the cytoplasm. It's a pox virus. Ever heard about the mpox, the disease? I don't know why they changed it, because the virus is called monkeypox virus. One of the pox viruses, it's like a bull in a china shop. It rams into the cell, absolutely doesn't care about the cellular immunity, breaks down everything, destroys the cell, and comes out. As one guy who is a prominent pox virologist, he said that the pox virus infection is the worst thing that can happen to a human cell. Okay? Eventually, virus particles get assembled, and then they come out in the process of release. Okay? Yeah. If the virus is naked, it will just pop through cell membrane. So usually, naked viruses lyse cells faster than enveloped, because enveloped viruses, despite the fact that they steal a piece of the viral membrane, the cell can quickly recover, kind of fuse it behind them. And sometimes these membranes come not even from the plasma membrane, but from like Golgi or endoplasmic reticulum. Okay? So I couldn't fit all of those considerations on the previous slide, so I got it in here. And the last thing that I want to discuss with you is the growth. Unlike bacteria, viruses have this eclipse phase. Hear me out. So first of all, viruses need cells. Everybody agrees with that? So let's say I have a layer of cells, and I add virus to it, and it attaches to the cells. And I keep sampling to see what happens in the fluid on top. Right? So I see some virus. Then I wash off the virus that didn't bind, and I start checking, and I replenish the medium over the cells, and I start checking the viral particles in the medium. There are none, because virus got into the cells and it is replicating there. This is the eclipse. When they're sitting inside the cell, accumulating particles. And then eventually it starts to come out. You start detecting it. Does that make sense? And at some point, the number of particles goes on the plateau. Why? Yeah, virus kills the host cells. Does that make sense? Yes? So why I pay so much attention to this? First of all, knowing the intricacies of the replication cycle allows you to make antivirals. Okay? That's how all antivirals came from the fundamental research. Anti-hepatitis C drugs, anti-COVID drugs, anti-HIV drugs, anti-hepatitis B drugs, all of that fundamental research into the functions and structures of viral proteins. And what was the second thing? Another thing kind of to keep in mind. Having cells in culture and having organism that is infected by viruses are two very different things. Does that make sense? So you can have a virus that infects humans with extreme success, like human papillomavirus, HPV. But when you start growing it in the cells, in culture, it sucks. It so poorly grows. And the growth is essential for preparing vaccines, for example. Because if you can't accumulate enough virus, that may be an impediment to the vaccine development. Not always, but can be. Does that make sense? So we will continue this conversation. So the first virus, quite appropriately selected, I think, considering the season, is influenza virus. So first of all, structurally, what we are looking at here is the negative single-stranded RNA. Now, structurally, this virus contains an envelope, and as we discussed, 2 weeks ago, which is like a long time, this virus is not particularly stable. So if you're concerned about, for instance, your child's toys being contaminated with influenza after they had the disease, just stick them in a box and put them somewhere in the storage, close it for 3-4 days, and the virus will disintegrate on its own. It's not particularly stable. Now, as you all know, there are a lot of different strains of influenza. To be scientifically exact, we're talking about influenza A. Main causes of human disease are influenza A, and to the lesser extent, influenza B, while there's also influenza C. In our conversation, I'm going to focus pretty much entirely on influenza A. By the way, symptoms aren't different. The disease is the same. It's the classification issue. So influenza A is classified based on the surface proteins, hemagglutinin and neuraminidase. So hemagglutinin in this image represented as kind of a blue buttons that have a stalk and the surface part. Neuraminidase is the enzyme. Its activity is required for the virus to leave the cell, and we will talk how it happens. Hemagglutinin has 16 subtypes, and neuraminidase is 9. So this is where this terminology, such as H1N1, comes from. When you hear that there is an H1N1 influenza virus, it means that its envelope contains hemagglutinin type 1 and neuraminidase type 1. Now, not all strains of influenza can infect humans. Obviously, you don't need to memorize, which ones can. What I want to talk about instead is the transmission. First of all, transmission between humans is respiratory, right? That's it. It involves not only sneezing or coughing, but also activities like talking or singing, anything that will generate respiratory droplets. Now, to put the end to the absurd discussion whether masks help and the argument that viruses are so small that they're not contained by the mask, the goal of a mask is not to contain a virus. The goal of a mask is to contain respiratory droplets. that are large enough to not go through the mask. Okay? They do help if you use them properly. Influenza is a zoonotic infection. So new strains of influenza virus usually go through the following steps. It starts in birds. For the most part, for human virus, it starts in various waterfowl. The feces, and by the way, in waterfowl, it's a GI disease. Okay? So when fecal matter from the waterfowl ends up around pigs, pigs get infected, and in them the disease becomes respiratory. That transition through pigs is essential for making an avian influenza, the bird flu, into a human one. Now, influenza infects a bunch of species. For instance, there's a big problem now somewhere in South America, I think. They have a big colony of some king seals or something, like some seals. And they are getting infected with, where are my seals? H7N7. And it just blows through the population and they die, and it's really a bad thing. Okay? Now, before we get into nitty-gritty of treatment and vaccination and why it's so hard to get vaccine working, I want to address the problem of a bird flu. Okay? So sometimes the infection, the virus, can be passed directly from waterfowl or any other birds, such as chickens or turkeys, to humans. Okay? And the thing is, bird receptor, the receptor for the avian influenza, alpha-2,3 sialic acid, isn't the same as the receptor for the human influenza. So in birds, virus binds to alpha-2,3 sialic acid, while in humans, it binds to alpha-2,6 sialic acid. Please do not memorize these terms. Okay? Partially because I just can't. I always mix them up. I can never remember which one is which, and neither should you. But here's the point. Humans have the same alpha-2,3 sialic acid in the lower respiratory tract, which means that to get infected with the bird flu, you have to inhale aerosolized bird feces. You can do it if you work in a big bird farm, like chicken farm. For this, you have to take off your PPE, you have to make a deep breath to deliver it all the way down into the bronchi and lungs, and then you can get infected. Okay? It can be very serious, it can cause pneumonia, it can kill people, sure. But here comes an interesting conundrum for the virus. While the person who is sick will be coughing and spreading the virus around, other people cannot get infected because human influenza is the upper respiratory tract infection. When you inhale those respiratory droplets, they end up in your nose and your throat, where you have the wrong receptor. Does that make sense? And it's been excessively shown that direct transmission of avian influenza from person to person, I'm not saying is impossible. I'm saying that it hasn't been observed. One of the issues that we are facing now, turns out avian influenza can infect cows. And the cow bird flu can infect humans. So when it passages through cows, we can get the bird flu that went through a cow and get sick. Is it more virulent? There's not enough data. For now, it appears it isn't. By the way, if you're curious, if cows get sick, they do get sick. They produce less milk. So, it's not good. All right. Do we need to talk about the symptoms? How you feel when you have a flu? I think everybody knows. Prevention. Hygiene. Avoiding contact with people. Treatment. Not really great. Amantadine. Pretty much all viruses developed resistance to it. Relenza and Tamiflu. If they're given in 24 hours after the onset of symptoms, they reduce, on average, the length of symptomatic influenza by 2 days. That's it. They do reduce mortality, significantly reduce mortality, okay? Now, more and more influenza strains become resistant to those drugs because we use them quite, ad lib. There's a new drug called Xofluza. I don't remember what it targets, but it's pretty much the same efficiency. And there will be resistance eventually. So how big of a problem is influenza? I'm going to give you some numbers. So the estimate for the last flu season, which was fairly bad, is 60,000 deaths, 300 pediatric deaths, and if you think that those 290, okay, 290. Out of those 290 kids, practically all of them had no underlying conditions, so they were otherwise healthy, and virtually all of them were not vaccinated. Which brings us to the idea of the flu vaccine. And is it good, and should you take it? So first of all, is it good by the same standards that we apply to all other vaccines? The answer is no. And we will touch upon why in a few minutes. Should you take it? Absolutely. Because the flu vaccine will not prevent you from getting disease. And I mean disease, not just infection, disease, actual disease. You can get sick. But it significantly reduces your chances of ending up in the hospital and or dying. Okay? It is really efficient in doing that. So why the vaccine is such a problem? So, 2 factors. First factor is high rate of mutations. It's an RNA virus, so it naturally has high rate of mutations. We call this high rate of mutations antigenic drift, implying that the virus changes ever so slightly every year, but changes enough to avoid being detected by the antibodies of your immune system. In which part of the virus are we specifically talking about? The head of hemagglutinin. This is the part that is essential for binding to the receptor, and this is the part that the vaccine raises antibodies against. So imagine that you take a vaccine this year. Next year, the head of hemagglutinin is slightly different, and that's enough to reduce the efficiency of your last year's shot to basically negligible numbers. Okay? So what can we do about that? I remember when during Obama presidency, the US government introduced the initiative for developing universal influenza vaccine in 3 or 5 years. And I was laughing my brains out because nobody understood. No, it's not going to happen. And the idea of a single shot transformed to the idea, how about we give a single shot but every 5 years, to how about we give a single shot every year but it's more efficient than what we have now. So one of the challenges is that your immune system prefers to make antibodies against the head of hemagglutinin rather than, for example, a stalk. And the interesting thing is that the stalk doesn't change as much. Right? So it would be great to make a vaccine against that targets, produces antibodies against the stalk, but it's really hard to do that. Now, how do we get influenza pandemics? I don't know how many of you remember, some of you weren't particularly concerned about influenza 16 years ago, I presume. In 2009, was an influenza pandemic caused by a virus that was a recombination of 3 other viruses, one swine, I believe it's 1 swine and 2 bird viruses, that ended up literally in the same pig. How does that work? So it turns out that the genome of influenza isn't a single RNA molecule. It's 8 RNA fragments. So imagine that Sean and I have 2 stacks of cards. Each stack has 8 cards. Sean has 8 cards that are all spades, and I have 8 cards that are all diamonds. So we are 2 viruses, effectively. We put our stacks together, right? Shuffle them and deal 8 pieces to each other. So now we both have spades and diamonds. This is called antigenic shift, the reassortment of the viral genome fragments. When this happens, the virus that arises usually has no resemblance to viruses that the population saw before. It doesn't necessarily mean that this virus is super deadly, okay? So far, doing all right? So in 2009, exactly that happened. The population wasn't immune. There was a wave of virus infections. It was not virulent, any more virulent than the regular influenza, okay? So if you wonder what next pandemic is going to be, probably flu. So another group of RNA viruses, this time viruses with a positive single-stranded RNA genome, are flaviviruses, named so because the first flavivirus that was discovered was the yellow fever virus, and in Latin flavus is yellow. So we're going to talk about several of these viruses. So the viruses that I highlight with yellow, West Nile, Japanese encephalitis, and tick-borne encephalitis, all cause, you'd be surprised, encephalitis. The pink ones, dengue and yellow fever, they cause hemorrhagic fever. Okay, we're going to discuss what this is. Also, interestingly enough, the yellow viruses cannot be directly transmitted from person to person. They come through various arthropods. The most enigmatic, of course, is tick-borne encephalitis. You will never guess which arthropod transmits tick-borne encephalitis, right? It's a tick. Japanese encephalitis and West Nile are transmitted by mosquitoes. But mosquitoes or ticks cannot transmit the virus by biting one infected person and then another uninfected one, right? Dengue and West Nile can be easily transmitted in the urban settings by mosquitoes from human to human. So far, doing all right? Now, the list of flaviviruses isn't exhaustive here. I'm showing you the most common, important. I wouldn't, like, tick-borne encephalitis, like, it's not a concern in the States. There's none. It's a pretty much a concern for Europe. Japanese encephalitis, Japan and China and Southeast Asia. West Nile dengue and yellow fever. This map that shows you yellow fever distribution, if you extend it over here, that would cover dengue as well. Again, West Nile, based on the name, you can imagine where it came from. It was first isolated in Africa. But now it's like all over the world. It's in Europe, it's in the United States, it's everywhere. Okay? So what does the disease look like? All of those diseases, all of them, all these 5 diseases are biphasic. For some reason, I singled out yellow fever and dengue, but what does that mean, biphasic? When people get infected, they develop what virology textbooks describe as flu-like symptoms. My former scientific boss in Louisiana had a chance to be on the plane with the American military serviceman who had dengue while being deployed. I still remember that quote that she mentioned. He said that if he could physically reach for the gun, he would shoot himself. Dengue means break-bone fever. You have 206 bones in your body, and every one of them hurts, and every joint hurts. And you have fever that runs like over 104. And you're going to recover on your own. There's no treatment, just, painkillers and antipyretics, and you're going to recover. And most patients, for most of them, that's going to be the end of it. Period. That's it. And you're immune. Some folks, though, will develop second phase, which, in case of West Nile, tick-borne encephalitis, or Japanese encephalitis, is, encephalitis, brain infection, inflammation. In case of dengue and yellow fever, it's hemorrhage. And in case of dengue, during hemorrhage, people can develop hypovolemic shock because it's not like they're bleeding out of the ears and noses and eyes. It means that they start losing fluids out of the circulatory system and the tissues. Decreased blood volume, hypovolemic shock. Okay, so what can we do about this? Well, I mentioned no treatment except for supportive. Can we vaccinate? Yes. There's a vaccine against tick-borne. I'm vaccinated since I worked with it. Japanese encephalitis. Yellow fever. I'm vaccinated because I worked with it. Went swimmingly. One of the best vaccines ever. But there are no vaccines. There's no vaccine period against West Nile and for humans. And there was a vaccine for humans against dengue, but dengue is a very complicated virus immunologically. So I'm going to make a very long and interesting story short. The vaccines have been recalled, and now there are attempts to make a new one that is better. Let's put it this way. Anyone owns a horse. In the United States, one of the biggest fears of the horse owners is West Nile virus. There is a West Nile vaccine for horses. I bet it would work for humans, but nobody tested. Because we have two-digit, lower double-digit number of cases a year. Nobody cares, Does that make sense? And the flavivirus that gained some notoriety in 2014, 15? Yeah, kind of 2015, is Zika. I don't know why I isolated, because Zika is not in the news anymore. But it was a pretty interesting story showing how epidemiology works. So I'm going to try to be fairly brief. It all started, it was isolated in Zika forest in Uganda. This is perhaps my favorite way to isolate a virus, by using a sentinel monkey. And if you imagine a monkey somewhere up there being a sentinel, you're not too far from truth. They placed the monkey in a cage and hung it from a tree. And just not monkey, the cage. And waited for the monkey to be bitten by mosquitoes and get infected. And it was, and they isolated, blah, blah, blah. Eventually it spread through Africa, then it spread to the Southeast Asia. Actually, retrospectively, it was found in patient samples. And then it spread to South America, most likely during the travel, I think it was World Cup or something like that, or Olympics. It was something. Yeah, for some reason I think it's, I don't know, some event. And then the shit show started. So the symptoms of Zika virus infection are actually pretty mild. So rash, conjunctivitis, joint pain, muscle pain. It's like, you're pretty sick. It's kind of a dengue light, okay? People were not particularly concerned until the group of physicians sounded the alarm that they observe a lot of microcephalic births among women who had Zika virus infection during pregnancy. I was, and I still am, an avid listener of my favorite Virology podcast, and they were kind of tracking down the development, and it was captivating. So think about the conundrum that you're facing. So you have a bunch of pregnant women, right, that were infected with Zika virus. Some of them give birth to a kid with microcephaly, some of them don't. It's definitely more than in general population. So you definitely have an association, but how do you establish the causation? It took a special mouse model, and the Slovenian woman who caught Zika while working in South America as a volunteer returned back home and had a miscarriage, well, stillbirth. It's not a miscarriage, it's a stillbirth, and donated the stillbirth to scientists. So they actually showed that the brain pathologies that they see in microcephalic, in kids with microcephaly, those brain abnormalities are co-localized with Zika virus infection of the brain. So it took several months of very intense work to end the application of special criteria, Shepherd's and Bedford Hill, to establish that this virus significantly increases the risks for the microcephalic birth. And actually it was shown that kids who were born apparently healthy had moderate, not severe, but developmental delays all the time. Okay? So now it's not really, I don't want to say it's not a big deal, but pretty much every person in South America got infected, and it's a lifelong immunity, and it's better. Now, if you think about, oh crap, I'm going to travel to, I don't know, Mexico, and I don't want any mosquitoes to give me Zika virus, you won't get it because they spray heavily. It's not like, unless you go into the jungles. Okay, but no, seriously. Yeah, and another thing to consider, if you're female, think, am I planning to have a baby? If the answer is not really, Go. Yes, I am. Well, go and then wait for about half a year. Interestingly enough, if you mail, same idea, because there are several cases, by several I mean like 4, of sexual transmission of Zika virus. Yeah. It's an anomaly. It ended up in the testes, because testes are immunoprivileged site, and then ended up in sperm. Somehow, it's super rare. It's like, there are several people in the United States that drown in the soup every year. Like, yeah, it shouldn't concern you as a potential way of dying, okay? Polio. I apologize, folks. Whenever I start making the slides about viruses, I go absolutely bananas. So I'm going to highlight the most important features of each virus. First of all, it's another positive single-stranded RNA virus. Those are the best viruses, and it's not an opinion, it is a scientific fact. Relatively small, non-enveloped. And that makes this virus pretty resistant. And it can persist in the environment for quite a long time. We can skip this entire replication thing because I feel kind of embarrassing to put it there. It's a material for like Field's Virology textbook rather than the general lecture on viruses. So I'm going to jump to the transmission. Its reservoir is soil and water. Basically, humans pass it in feces, and water treatment plants aren't aggressive enough to inactivate virus. You can actually detect poliovirus in the sewage. It gets in the drinking water, or just water in the pools and stuff, and infects people via, oral route, replicates in the gut, and then is excreted in feces. Usually, your symptoms in case of polio infection are limited to fever. Okay? Does it make sense? But in a very small amount of folks, 1 out of 200 to 100 cases, so 0.5% to 1% chance that there will be a spillover, the virus will get out of the gut and infect the neurons, and then move through the neuron all the way to the anterior horn, the spinal cord. Those neurons that sitting in the anterior horn are motor neurons. So this infection will lead to flaccid paralysis, meaning you can't contract the muscles. If you think where your gut is, it is absolutely conceivable that these kids have paralysis of the legs. Right? That's the horns that are affected. In some cases, the virus can climb up and paralyze the diaphragm, which results in the respiratory arrest. And people with paralyzed diaphragm had to live in iron lung. These machines effectively breathe for you. They expand your chest. They replace the respiratory muscles. Does anyone know how many such machines are left in the world? At least in the United States. 1. There's 1 machine. The last person who actively used iron lung died a couple of years ago, and he was pretty successful New York lawyer. Lived until, You don't have to live in it. We have a lot of respiratory muscles. We have diaphragm, we have intercostals, we have levator scapulae, we have scalenes, okay? Accessory muscles. So, when you are conscious during the day, you can use those muscles to breathe. It gets tiring, you get an iron lung, take a break, come back out, you spend the night in the iron lung. Okay? So,
Can we reuse that same machine after a person dies?
Yeah.
That guy, the one guy, seemed like he was in there for night and day. The last one that you're thinking about?
Maybe because he was like old already,
Yeah.
because respiratory function decreases with age. But technically, like, if you ever had a really bad asthma attack and you had to produce labored breathing to push or pull the air in, you can do that. It is tiring. As a person who had such attack, I can tell you. Okay? There is no treatment, but there's a vaccine. There are two vaccines, actually. One is inactivated, developed by Jonas Salk. If you were born in the United States, you received inactivated polio vaccine. Okay. If you were born in the United States after 1953, which I presume you all were born, yes. Now, what about if you were not born in the United States? Yep, that's Sabin vaccine. So Albert Sabin developed an oral attenuated vaccine, which means the virus actually replicates in your gut. Okay? And basically, he ran one of the largest clinical trials because from the government, US government standpoint, Sabin's vaccine was useless because we already have Salk. So Sabin was a good friend with a Russian virologist, Mikhail Chumakov. I actually did my PhD in the research institute named after him, the institute that he founded. Okay? Like a really cool guy. And asked him, how about, you need a vaccine, I have a vaccine. It's not super thoroughly tested, but it is tested. How about I'm going to give it to you? So Chumakov built an institute, they built an institute to produce this vaccine, made a bunch of it, vaccinated folks in the USSR. All 200 whatever million. Okay? So, it seems like the immunity that attenuated oral vaccine provides is more robust. But let's talk about the advantages and disadvantages of both. So, inactivated vaccine. What are the pluses? Safety. Like, there are no side effects, period. It's effective. It 100% protects against the polio disease, not infection. Disadvantage, requires cold chain storage issues. And I have a question for you. Can I give somebody an injection legally?
Can you?
Yeah, me, right now. No, because I'm not a medical professional, right? So it requires a trained medical professional and ideally a proper environment, okay? Now, what about live vaccine? First of all, it's cheaper, significantly cheaper. Second, I don't remember the administration of mine because I was, 3 months old? But I remember my kids being vaccinated. It's a drop on the tongue, and you swallow it. I see Richard is nodding. Yeah, that's it. Who can administer that? Anyone. You know which country ran the largest polio vaccination campaign recently? India. I don't remember the exact numbers. We're talking hundreds, plural, of million people. They had volunteers just door to door canvassing, and they had a chalk with them. They went into a household, vaccinated everybody, then put a sign on the door, like vaccinated for other folks so they don't vaccinate twice. What's the problem then? Like, it sounds awesome, right? So the problem is this. The strain that is attenuated, the weak one, any strain actually, not only type 1, type 2 does it, 2 type 3 does it. When they go through the gut, they revert from the vaccine, the attenuated type, to wild. Now, at this point, you're usually already immune, so you're fine, right? But what if you're not? So 1 out of every million vaccinated, on average, developed vaccine-associated polio. At the time, think about the challenge of vaccinating, 240 million people very quickly when you don't have the industry to make an inactivated vaccine. Well, let's do this with live vaccine. The cost was considered acceptable. Okay? By the way, little note, do you think that these vaccines are patented? They're not. When Salk was asked if he's going to patent the vaccine, his answer was, would you patent the sun? So he gave it, the problem in Soviet Union was it didn't have an industry to make it. Live vaccine is much easier to make. So you can see that mass vaccination campaign in the States eliminated polio. We're still free, the US is free of wild poliovirus cases, which brings me to this wonderful map here. So this is an old map, I acknowledge that, but it's a really cool map. So the orange circles here in Central Africa and in Southeast Asia are vaccine-associated polio infections because those folks received oral vaccine. Okay? It's inevitable. And now think about this from another perspective. When you get an oral vaccine, hear me out, it's a virus which infects you. And then you shed it in the feces and it ends up in the environment. Are you still with me? It's pretty stable in the environment, and it can infect from the environment another person who hasn't been vaccinated yet and cause the disease in that person. For instance, last year, there were few cases of vaccine-associated polio in Nassau County in New Jersey. A couple of unvaccinated individuals came to states. There is a huge, I don't want to sound culturally insensitive, so Indian, probably Bangladesh, Nepalese population in New Jersey, okay? The second largest Hindu temple outside, the largest Hindu temple outside of India is in New Jersey. If you haven't been, you should. It's beautiful. It really is.
You've been there?
Yeah, it's wonderful. So they got infected because folks who came from India and were vaccinated were still shedding the virus, and it was in the sewage, and it somehow got into the water, and they drank that water, but they were not vaccinated. Voila. Does that make sense? Unfortunately, the only way to prevent those vaccine-associated polio cases is to switch to IPV, inactivated polio vaccine, which is hard because it's expensive and requires a lot of logistical adjustments. Now, what about this? This is at the time, the border between northern Pakistan and South Afghanistan. That year, 2018, it was a territory that was controlled by Taliban. And they decided that people who were volunteering to distribute the vaccine locally work for the US government, which some of them probably did, but not in the way that Taliban was concerned. They started killing them. And the volunteers said, screw that. and got out. So this is the cluster of wild polio. Does that make sense? Not vaccine associated. This is the only place on Earth right now that has a transmission of wild poliovirus. Okay? Now, I have a couple more. Then we're going to take a break, and I'm going to arrange all that stuff here and start, chatting about protozoan helminths. Hep C. Now listen, I'm talking about hepatitis C. Hepatitis B is fairly similar. So I'm going to be mentioning it from time to time, but since it's not in the notes, it's not going to be on the exam. So hepatitis C. Distant cousin of yellow fever, interestingly enough. It is positive single-stranded RNA virus. It is enveloped, so not particularly stable. And it is very hard to work with. Let me explain. Human hepatitis C virus infects humans and primates. Can you work on primates in the United States? You can. You can't work on apes. Also, primate work is insanely expensive, thousands of dollars per monkey. So it's hard, okay? The cell culture system, because viruses require a host, took 10 years to develop, okay? So it took a lot of time to develop it. And since there is no convenient cell culture system, no convenient mouse model or animal model, whatever, it's really hard to do the vaccine. Okay, so how is it transmitted? Needle sticks. So what is the population that is affected the most? Tell me. What? People who use injectable drugs. What is the best way to prevent hepatitis C transmission among the users of injectable drugs?
Clean needles.
Clean needles. Have you heard about a gentleman named Mike Pence?
Yeah.
He decided that the best way to stop the drug use is to not give people needles. When he was a governor of Indiana, and Indiana saw a 70% increase in HIV and hepatitis C infection rates. Okay? Yeah. By the way, Russian politicians did that same thing and saw the same results. So politicians are idiots. So what happens to the liver? That happens in case of both Hep C and Hep B. And hepatitis B is also transmitted. They actually very similar in transmission. So needles, right? Both Hep C and Hep B. Sexual transmission, not as easy. Okay, can be, but not as easy. And vertical transmission from mother to child. If you are practicing safe sex, don't use injectable drugs, and aren't born to a Hep C or Hep B positive mother, can you still get this infection? How?
Yeah.
How?
Sorry, by exposure to blood.
Well, I'm not, you're super careful. Like, no needle sticks, no syringes around you.
Breastfeeding.
I mean, can you imagine me being breastfed? Like, no. No, all blood is tested. Every batch of blood is tested for Hep C and Hep B. Think about needles that people voluntarily expose themselves to. And I'm not talking about kinky stuff.
Accidental sticks?
Huh?
Accidental sticks at work?
No, not. We saying no syringes inside.
It's not.
Huh?
So I guess not.
Say again?
I said, I guess not then.
I'm giving you a hint.
Oh, tattoo.
Yes.
Oh.
If you're using a funky tattoo master and they're not really particular about hygiene, yeah. Okay? So you see the numbers aren't too bad. Actually, in the United States, we're doing all right on hepatitis C. Okay? So we have few states. So think about this. Green isn't great. Blue and dark blue is good. Okay? Now, we don't have and what happens? In both cases for hep C and hep B, your liver gets inflamed. Some people, by the way, clear the virus completely. They get infected and then they not. That's it. Some people develop inflammation, some people develop cirrhosis, and about a quarter develop liver cancer. So what can you do about hep C? There's a treatment. Last year, I think it was last year, some recent years, 3 scientists, I think 2 American and 1 British, received the Nobel Prize in Physiology and Medicine for the development of hepatitis C treatment. And it was and by the way, they just studied fundamental properties of the virus. How virus works, how the enzymes work. And based on this, developed drugs, which leads to 95% recovery. Now, you can never be considered free of hepatitis C, but it's undetectable and long-term undetectable. So basically, you clear. Okay? Now, I really need to check how much it costs now out of pocket. But in 2019, 2020, that was the price for Gilead course. $84,000 for 12-week course. $1,000 a day. A bit prohibitive. Okay? Well, hepatitis B, there is a treatment which isn't super good. But what's important, there's a vaccine. And it's one of the best vaccines developed by humans. It pretty much prevents an infection. Does anyone know when it is given? Like if a child is vaccinated, how early?
When they're like one.
What is the first vaccine?
One what?
1.5.
1 day.
1 day.
It's given after birth. It has 0 side effects. And it has to be given in 3 shots. A child has very little authority over their decision making, so the parents can bring them easily. And they're not really busy, you know? Like as an adult, I started my Hep B vaccination. I got my first 2 shots, and then life got in the way, and I missed my 3rd, and I completely forgot, and I don't know. No, I don't know what to do. Okay, so probably they're going to restart. Another issue is the only issue with Hep B vaccine, it's expensive. Not every plan covers it, which is absurd if you think about it from the perspective of a health insurance company, because they should cover all the vaccinations. It's much cheaper than to cover the hospital stay. Okay, so, and by the way, I think that's what they did with the COVID vaccine after the CDC removed that it's recommended. Morons. Okay. Norovirus. It's very timely, folks. When I was driving, I was listening to YouTube. I insist. I wasn't watching. I was listening. And the host of the show was talking about the spike of violent vomiting disease, also known as winter vomiting bug, or 2-bucket disease.
Yeah, it's actually going around really bad. I had about at least 6 patients last night that had
How many?
6 out of my 24 beds. Yeah.
What for?
It's 24 beds
Okay.
6 or 7 of them.
So how old were the patients? If it's not like a HIPAA violation. Ballpark, I don't need age.
Between like 50s and 80s, 50 to age 80.
So I switched up, I'm going to go back for a second, but 20 million cases a year. Just cases. That many hospitalizations. Very few deaths compared to, how many people get infected. Those who die, older people, and infants and toddlers. Because they often cannot hydrate themselves properly. That's it. Okay? So, what does it look like? Like a two-bucket disease. You barf and shit for a day or two, and then you're fine. Okay? Here's the tricky part. I have plans to ruin some food for you today, but I'm going to ruin the restaurant industry for you right now. This disease can be transmitted during incubation period. Remember what incubation period is. You have no symptoms. It can be transmitted, obviously, when people are sick through the aerosolization of vomitus or aerosolization of fecal matter. And it can be transmitted during the convalescent period. So next time you are eating at a restaurant, look at your waiter and ask yourself, is this person in the incubation period of norovirus? Well, maybe they recovered a week ago and still can spread. And then think about the hand hygiene of an average person. The infectious dose is extremely low. To illustrate that, I'm going to tell you a real case from one of the US universities. I can't remember which one. So they had a chili, like big party with a big chili cook or whatever. So they had a big, like a barrel of chili, like really big. And some student who, for some reason, wanted to exact revenge on, prank, I don't know. He got in the room and took a dump in chili. What do you mean? It was a good chili. Nobody felt anything. It was really spicy. So it was pretty diluted dump at this point, right? It's a huge barrel. So people ate this chili despite of the heat and dilution. Everyone got sick.
No way.
I can tell you that we had an experience when our friends told us they wanted to visit us. And one of them told us that, I recently just recovered from it. Are you okay with that? And I go, ask my wife, do you really want to see them? She goes, yeah. Like that much? She goes, yeah. Okay. We paid a price. It's basically, if you hang out with a person that sheds this virus, you're going to get sick. Okay? Period. It sucks, but, so first of all, why is it winter vomiting bug? Nobody really knows, but most likely it's because people are confined, inside. And who's affected the most? People who go to school and people who are in the nursing homes. What they have in common? Poor hygiene. I mean, seriously, did you see kids? Now, I want to tell you a sort of an interesting story how it was studied. So when it was discovered, by the way, it was discovered in Ohio. It was discovered in the city of Norwalk and was named originally Norwalk virus, which made people of Norwalk very displeased. And they asked to rename it. This is why it's called norovirus. And turns out human norovirus cannot reproduce in any other animal besides humans. So until the discovery of mouse norovirus, which does similar thing to mice, the only difference, mice do not have vomiting reflex. They just can't vomit, so it's only diarrhea. But how do you think norovirus was studied before the model was developed, like human norovirus? Who were the lab rats, in other words? Humans. And which humans are willing to do almost anything for a small compensation? College students, more specifically medical students. Because the explanation, the research is done, a lot of virology research is done at medical centers, and these are the most available students around. So students would get paid $300 to $400, would drink fecal suspension on Friday, have an unforgettable weekend, and go back to classes on Monday. The person who told us that whole arrangement, she's a world-class researcher in norovirus. She said that allegedly in one of the schools, students were rewarded with not with money, with extra credit. And I've heard reactions like, yeah, if it means, get an A, I do it. Yeah, if it's an A, why not, Okay, does that make sense?
Don't apply to us.
I'm not doing any research like that, so don't worry about that.
Oh, man.
You sure?
Say again?
You not doing any like research studies on this?
No. But first of all, what I want you to take home, it does not induce a long-lasting immunity in the infection. So you can get infected and then get infected with a different genotype like 2 months later. You can get infected with the same genotype 6 months later. Second, it's not that bad, really. Electrolytes, 2 days, you're fine. The good news, the vaccine is in the trials and it shows pretty good efficacy. So we may be spared, okay? Now, before we take a little break, I'm going to highlight what you need to know about protozoa, because I need to do this to free some space. What do we call protozoa? Single-celled eukaryotes. That's the definition. Always one cell, always eukaryotic. No cell wall, by definition. 2 layers, ectoplasm, outer layer, movement and feeding, endoplasm, all, cellular structures, regular things like nucleus and mitochondria and whatnot. Many protozoa can form cysts. So hear me out. The living and feeding form is called a trophozoite. When the environment becomes kind of shitty, trophozoite forms a cyst. Cysts do not reproduce, they do not feed, they just hang out. And then cysts can infect other host. Okay? So, this is what I'm talking about. So you have a trophozoite, you have cyst. They go like this. So bad environment becomes a cyst. Good environment goes back to become trophozoite. End classification, so we're going to take a break and then start next lecture on the different species of protozoa. They classified based on locomotion. So pseudopodia, sarcodina, or amoeba, obviously, all move by producing, pseudopodia, like amoeboids. Mastigophora move using flagella. Cilia is used by Ciliophora. Now, if you look at these 3 groups, the groups that can move around, all of them have free-living organisms, organisms that do not depend on another host to survive, because they can move and chase the prey. Apicomplexa, or sporozoa, do not have an ability to move. This is why they have to be parasites. Okay? Sounds good? Now, let's take a